But if there's no other updates and announcements, we will get into the meeting for today. Like I said, there's a couple of things on the agenda. So first up, we have a presentation from Courtney around the gene curation SOP updates. And then once that's complete, we're going to be moving on to finishing off the variant curation. I was just muted. We're going to be finishing off the variant curation Q&A session that we started at the beginning of September. We only got about halfway through that, so we've still got a number of people's fabulous questions to get through. But I will hand over to Courtney first up to give the SOP updates.
Great, thank you very much, Alicia. I'll just ask everyone if you can please mute yourself for now as we go over it, but I'm happy to answer questions. Please also utilize the chat, and I will do my best, as well as my colleagues, to look at that and get your questions. I'm going to go over and share my screen. I think I'm sharing my screen. No, I'm not. Sorry. There we go. Yes. Can you see SOP version 10 updates? Okay, yeah, great. So let's go ahead and get through this. And I'm just happy to present this on behalf of the Gene Curation Working Group. So we're going to go through several of the updates we're making for the SOP version 10 for the Gene-Disease Validity Curation. Each of these will go through on the subsequent slides. So first, we want to say that we have a title update. Previous times of the SOP, this was referred to as Gene Clinical Validity. In order to match what we do and advertise on ClinGen, we have changed it to the Gene-Disease Validity Curation process. So we just wanted to note that you may see that change on the title. Going through the next sections, as you come through the SOP, we do have a required components section. And so we did add updates and reference links for both the lumping and splitting publication. This especially concerns the process of pre-curation that our gene curation expert panel biocurators, as well as the panel itself, go through. But also we've had the new release of the disease naming guidance out of the Disease Naming Advisory Committee for ClinGen. So we provided a link to that. I also want to mention that if you all know where the lumping and splitting working group pages, we have co-links back again to the naming guidance. There's also a separate disease naming advisory committee under the working group tabs on the clinical genome website. But those should be in the SOP as well, so that they're handy for you if naming comes up in your particular call for your curation. Again, to the required components section, underneath the access to the scientific articles and publications, we did add a note about the use of preprint articles and the fact that there can be valid evidence within these articles. So certainly it is up to you as a group, and even as biocurators, we encourage you to look even out between PubMed to see whether there are any other pieces of evidence. In saying that, though, we do want to make sure that if it is a pre-publication, that you're really bringing this information to your gene curation expert panel to see if they are feeling comfortable with using and scoring that curation as information as part of your curation, especially wanting to score or include the classification there. We recognize and respect that these are pre-published. They haven't gone through a peer-reviewed process, and so some expert panels may not want to utilize this for the actual score classification. However, they may want to note about the existence of this data. If they do, we do encourage you to include information and make necessary remarks in your published evidence summary. If you do, in fact, want to use this information as part of your curation at this time, we would ask if it modifies the classification, you will have to use that function in the gene curation interface, which will require a rationale. Again, in that rationale section, please include the fact that you are using pre-published data, where it comes from, the reference, and if possible, a link to that data. When you're using bioRxiv and medRxiv, they do have stable links that are associated with a DOI. I do want to indicate we do not have the ability to support entering this information as you do with the PMID at this time, but we are working on this for pending in future efforts, again, using a DOI or another stable identifier. I'll also remark with this is that there is an effort right now between PubMed Med and bioRxiv, medRxiv to actually put PMIDs and abstracts for pre-published articles. So you may, in fact, actually find some that have a PMID. In that case, you also would want to remark that that is a preprint article. And of course, that PMID would have that attribution as well. As we move through the SOP and looking at the clinical validity classification examples, we did the examples that we have based on that final classification where needed. For instance, previously for moderate, we had a gene disease relationship of SCN1B with generalized epilepsy that was moderate. This was at last year's release in August of 2022, but you can see by December, this had moved up to definitive. So please just make sure to reference if you want to come back and look at these for what we have linked here, but also recognize they may be changing with time as they're being recurated. You can always go to the clinicalgenome.org website also, looking under all of the gene curations and picking by classification, moderate, limited, or whatever example you want. You would have the best way to track those examples if you want to see how they've been curated. We also want to remind you that pre-curation IDs are required to start a record. This is something we released this past summer, so all of you, I think, are well knowledgeable about it, but for anyone who's new, we just want to bring that back around. We did add this information into the SOP just to remind everyone and new curators that GCI and gene tracker records, in order to start a new record, do have to match in specific fields. Those fields include the HDNC ID number, the Mondo disease identifier, that Mondo ID, also the mode of inheritance. Each one of these uses human phenotype ontology terms to distinguish the mode of inheritance. And you also have to have the affiliation. So making sure that you are signed in the GCI under the affiliation for that record. It's the only way you can create a new record. But for those of you who may be on multiple gene curation expert panels, please make sure you've chosen the right affiliation. And then lastly, the pre-curation ID. You can see on the right here, this is the modal when you're in the gene curation interface, and when you have to enter all these pieces of information to match. An error will be given to you if one does not match. In this image, you can see that what doesn't match with this particular record between the ID is the disease. So you need to look back in the gene tracker or talk to your GCEP coordinator about what was actually chosen for that ID. And you'll get those different errors depending on if your error is in the HDNC ID or the mode of inheritance. We also wanted to provide some clarification on the use of the semi-dominant mode of inheritance. Our previous language really indicated that it had to be a dosage-dependent effect when it could be used, which means that you have a more severe earlier onset or some other type of change in the phenotypic feature severity. And in fact, we updated that to say it could also occur similarly between both monoallelic and biallelic observations of the same variant. So we wanted to bring a little bit more clarification around when this particular mode of inheritance could be used, and it doesn't strictly have to follow this more severe phenotype. This can be the typical, but it is not always what happens. Going on with mode of inheritance, we've had a lot of requests lately within our gene curation interface development team about wanting to change the mode of inheritance for a particular record. And so we wanted to provide clarity for you all in the SOP about this. In general, we will ask you to be very thoughtful and thorough, especially now having to have a pre-curation record for every gene curation to make sure that you have the right mode of inheritance when you are selecting that pre-curation and then into the gene curation record and the gene curation interface or GCI. It is very difficult to change this. It's not something we can change. A lot of the modals are tied to this with the scoring, but we do recognize there are a few that could update and change. So for instance, if you had an autosomal dominant mode of inheritance for your record, but realized you needed X-linked, we could change that and update it. The same we could do for X-linked to autosomal dominant and then mitochondrial. What you will notice here is that we cannot change recessive to dominant or X-linked. We can also not change semi-dominant to these. So again, please be very thoughtful about the MOI when you're choosing it to begin with. It could mean if you've chosen the wrong one, you can just start a new record with the correct one. I see there's a question about the mode of inheritance. You have a gene, so this is Joanna, that can be associated with disease in both autosomal dominant and recessive inheritance, depending on the variant. Which should I pick? Well, if it is associated in the variant and you go through your pre-curation and actually find it's part of a spectrum of a disease, that would be semi-dominant. So semi-dominant is where you can score both recessive and dominant or monoallelic and biallelic cases the same way. And you'd want to make sure that those have the same mechanism of disease, right? And they have similar phenotypic features, but again, they can vary or not by their severity and age of onset. If they are two distinct entities, you would want to create a record for each of them based on the mode of inheritance separately. And then you'd also want to separate the data that you're using, both cases and experimental, to make sure that they align with that mode of inheritance and that final gene disease relationship assertion. So great question, Joanna. Hopefully we got that answer for you. The other thing you will also see about this added into this table just for everyone and their clarity is what can actually be published to the website. There are uses of some, especially the other here, you will see that we actually cannot publish to the website. And even with undetermined mode of inheritance, we can publish to the website, but you cannot necessarily score any of this information because you would not know what the mode of inheritance is. In some cases, these may be able to change the MOI or not, and we can look into those in the future, but at this time, it cannot be changed. So we do want to talk about an area in the SOP that we have brought a little bit of prospective language into, and that's evidence collection beyond PMIDs. As remarked on about the pre-publications, we really are listening to you all as users saying that you want to have different pieces of actual published information that's out there to be able to use for curations. And so we are indicating that support for entering especially ClinVar SCVs is on the future horizon to use as evidence, and that you would be able to enter in an SCV much like you do a PMID and start scoring on cases. A really good example of this, and just to bring to everyone's attention again, clearly there are a lot of different genes and a lot of different entries into ClinVar, but our own ClinGen Genome Connect also publishes information on patients that have given their consent. These oftentimes have a lot of the clinical features and HPOs present, as well as their age of onset, and they have an SCV number. So if you happen to be doing a curation and find through Genome Connect that you have a particular patient in the future, we hope that you would be able to enter in this SCV number and curate on this particular variant and information that you have. Just as a reminder to all of you, you can go for any type of group into ClinGen into ClinVar and look at their submitter list. For Genome Connect, they happen to have a lot of different submissions over several different genes. So it's worthwhile to go look at that and see if there's additional information that you can use. So we'll now move into the genetic evidence section. Again, there's been no big changes. You all know we had a big change around SOP version 8 for our scoring, and we are staying consistently with that. We just wanted to bring some more clarification on that type of scoring for both monoallelic and biallelic variations. So you do know that a variant is going to be scored in each of those cases, and each variant is scored. If you happen to be on a biallelic or autosomal recessive, that's compound heterozygous, you'd have two different variants, and they would each be scored based on that variant. When you have your options in this drop-down menu, they have been expanded. So you can see the numbers here, but they're quarter points in increments after 0.1. So we do not have the ability to say 0.6, 0.8. However, depending on how you click your buttons together, you could get a 1.6 or a 0.6 calculated, because that's what the gene curation interface does. That's okay. We just wanted to make it clear with you, you would not be able to choose that particular score if you wanted to alter it. Again, for the autosomal recessive or biallelic conditions, a score is chosen from the drop-down menu that includes all of these, and then the two scores are added. Sometimes when you click that attribution of it does have variant level evidence, the GCI auto-calculates the 0.4 points to add on to it, as well as if you click de novo, it will add the additional set of points. So this allows for a calculation, but again, you can modify. If you're modifying, though, you are restricted to a certain number of points at this time. For this particular example, for numerical values, you can have 2 missense variants, right, with no supportive functional evidence or inheritance data, and that will be scored at 0.2 points, and it will stay there. It's not going to round up to the 0.25. However, if this is something your group wants and you want to change it, you are welcome to do so. We also updated the footnote just to refer to the types of rounding that happen within the gene curation interface. Going on, we wanted to provide some clarification around guidance and scoring for homozygous variants from consanguineous families, especially. So the language around this is considered downgrading due to the increased likelihood of identifying multiple homozygous variants in a consanguineous family, or something we refer to as runs of homozygosity. But that really is up to you and your gene curation expert panel. So we suggest, if you know and it has been asserted or outlined within your particular paper that there's consanguinity with a family, to talk to your GSEP about it. Ultimately, it's up to them how they want to score. We would just ask that you all keep this consistent among all of your curations across a gene curation expert panel. Also, we wanted to remind you that it's important to remember for homozygous variants, since there's only one variant, even if it's present on both alleles, you only indicate the score for the one variant. You do not need to add the variant again. The GCI will automatically double the points to reflect the presence of it being in a biallelic or homozygous case. So just wanting to clarify that for everyone. We've also added language about scoring copy number variants. This also was released more recently from our ClinGen allele registry. We have what is called a canonical allele copy number ID. That's a CACN ID here. This is what you would use if you have a copy number variant, and you can now enter these and score them in the gene curation interface. So we do provide you some examples and a link back to the ClinGen allele registry, so you can be able to access and get these CACN IDs. The process for this is still to choose the HGVS descriptor, and then you would go through and pick your chromosomal regions here. There were previous calls where we've reviewed this, I think back in April on Biocurator work group, so you can always hit the ClinGen YouTube channel for previous Biocurator work group to find how you can use and find CACN IDs. Excuse me, going back here, but one thing to always remember, which I'm sure you all do, but when you are looking at what are large regions, many times it could say in a paper that it's intragenic, but it is good for you to go check those breakpoints on the right transcript just to make sure there aren't any other genes or other non-coding regions included. If you do find there are other genes or regions, the ClinGen allele registry will show you that. You definitely want to bring that back to your group. That may mean you score this as 0, right? Points for that particular case. It's still curated, but it's not going to contribute to the final score. And this obviously will make it really clear, and the GCI will pull in that there are these additional genes. So you can add that to your rationale, and it will also be remarked on. Certainly it's at the discretion of your group if the paper ruled out by using different genes to show that they were not contributing to the effect, they could decide to score it. So just as in everything else, make sure you talk with your expert panel about the inclusion and scoring of certain data. So we did clarify language about the use of computational predictors here. I just wanted to bring into you the exact language. So where we say in silico predictions in general do not provide sufficient evidence for functional impact and are therefore not typically counted as supportive functional data. Obviously, again, you can talk with your expert panel about this. We do recognize that this guide is distinct from that made by the ClinGen sequence variant interpretation group that describes the role of in silico predictors, but this is in evaluating genes with established gene disease relationships. And this whole process of gene disease validity, you are working to establish it, and you may not know it will be strong or definitive to start with. So this is where we are giving that caveat of why we are not necessarily prescribing to using this as variant level evidence to score higher. Again, there may be expert panels that decide to reward the upgrade over the default, especially if it's particularly compelling in silico information. we would again just stress that you all have consistency across your expert panel and the genes that you're evaluating and how you score and use this particular type of data. We wouldn't want you could score it for one gene and not other, but really make sure you have some compelling evidence of why to do that. Otherwise, try to keep it consistent across all of them. We also wanted to clarify a little bit about what we mean as obligate carrier when we use that term within the SOP. So we've updated some language about this, removed past language, and we've provided a new figure for you. So obligate carriers can be those individuals that are between two affected individuals, even if they're not tested. This is especially true of X-linked conditions, right? That the female would be the one harboring it, and then it is between individuals where we have affected individuals at the end. But they can also be those individuals that have had the testing to show they're a carrier as well. Because we would not expect these individuals to actually present with any types of phenotypes. So we wanted to update some language too about segregation without a program. You can apply segregation scores to your gene disease validity curation and final classification. You obviously have the ability to associate a proban, and in most situations that might be what you're using. It's great. You can fill out all the information when it asks you a proban's name, etc. In the cases where you may have a paper where they were able to show lots of linkage, it's one gene area, but they had not been able to identify the variant responsible, right? In that or any subsequent paper, but they have a really nice LOD score to implicate that gene in the disease or the phenotype, and you want to use it, you can just leave all of the proban fields blank in that situation and say you do not want to choose a proban when prompted. When you look on the ClinGen website for the final gene disease validity classification, if you were to look under genetic evidence, there is a section that says case level family segregation without a proban, and this is what it would look like. So it has its own designated area. Again, that score is still applied to the segregation. The way that this looks different when we think about segregation without a proban versus with a proban, you'd be in case level segregation. And what you will notice is that we will associate the patient that's linked to that family underneath the proban family label. You do not have that in the upper region. It just says label only. If you made a mistake and you didn't fill any of those out and you have the segregation score and find, oh gosh, there is a proban that is associated with this particular family, you can go back and correct that. What you want to do is navigate to the dashboard for that particular record. You want to make sure you're selecting the PMID in which that paper and that segregation and proban came from. Navigate over to the right area where it says family, and then you want to, oh, this moved, but you want to go over to what the right segregation and family label is, and you can say add individual. So I apologize that this moved. You can also see that for this particular example, there was already a proban indicated by the number of variants associated for this particular family, where this is the one without. So again, this is the procedure if you happen to forget to put that proban label and want to go back. If you happen to create an individual first, you cannot then associate them back to a family. So please make sure when you start and you know that it's a proban with a family that you start adding the individual here. We cannot go back once there's the individual and associate with a family afterwards. Did anyone have a question, or did I happen to answer it? Okay, great. We also wanted to provide some clarification on the LOD score and how the gene curation interface, or GCI, auto calculates it. We did want to indicate that this particular formula that is used is assuming that we have a carrier mother and not an affected father, especially when we think of X-linked diseases. We provide this example that if it was the instance that the father was the carrier and passed it on to a daughter, your denominator would now be 1 because they had a 100% chance of getting that affected allele rather than 50%. So we do indicate that our estimated LOD, if you used it, is based on the formula written above of 0.5. If this happens to be different, I think you would have to just look at changing that estimated log or trying to figure the number of people that you have in segregations to alter it. We've also updated the case control data. In this, we describe new options for the case control data and how you can either score, contradict, or review. Previously, it was only score, but we realized for a number of gene curation expert panels that they were actually finding the lack of statistical significance or difference between a gene in cases versus controls could be used as contradictory evidence to dispute a specific gene-disease relationship. So you would have the ability to pick these two other areas or two other statuses aside from score. Those are translated to not only within the gene curation interface, but what we're looking at here is an example that's published on the ClinGen website. So that status score would indicate contradicts rather than a score, since you didn't find it as positive information. That means you don't have to score it anymore as 0 or things like that. You can really say contradicts when you feel that's what the data is exhibiting. And for the final summary and matrix section, we did just update the figure 9, again, based on the updates to the title to replace the gene disease association, because we prefer the word relationship or related rather than association. And we also want to let you know, we relocated the information about modifying a classification to this section because it worked better with the flow. So if any of your groups are considering modifying a classification, it is moved further down in the SOP, but you'll still be able to find it and understand what you need to do to use that particular function within the GCI. Also, a big thing that was just released that's really important, because we do want to make sure that we're putting forth an effort to version our gene curations. We recognize a lot of our gene curation expert panels are going through recuration processes and updating these versions. Not only that, we know that there are minor changes that need to be made because we're finding typos or other things. And so we really wanted to be able to attribute when there is a change to a record, what it was changed for, that it can help us to apply different digits to that version. And so we have called these curation reasons. They are available in the gene curation interface. They happen to be in the variant curation interface too, and they are very well aligned. So if any of you are on VSEPs, this will be important as you go and to publish a specific variant classification as well. These are required for all new curations that are being made. We're trying to backtrack for other curations and apply version numbers based on what we know and what we found through data analysis. You will be prompted before publishing to choose a curation reason. So if all you did was, I got assigned a curation by my GSEP, it's new curation. That's what it is if it's never been anything before, and you can move forward with that. But for those where there are recuration, or you find that you're updating a curation because you've made an administrative update, you can provide those rationale, and we understand you can have multiple. So for recuration and administrative, you could pick multiple of these rationale and reasonings. It's not necessarily going to give a whole bunch of numbers of versions, but we just want to understand why you were going back and making a change to that particular record. There's more clarification on this, which I'm going to go through in a minute, in our GCI VCI help document located in the GCI. So our Stanford development team for the gene curation interface nicely maintains a Git book and a wiki for the GCI and the VCI and help documents. And so they have clearly and nicely outlined this particular curation reason, when you will be prompted, and what it will look like as the modals within the gene curation interface. So if you happen to be in there and you are confused, you can always locate that help document. You will have info bubbles as well to tell you when you should choose these different ones and how the definition is for each of these curation reasons. So if you are confused, go ahead and look at those bubbles or link yourself back to the wiki document. We have a link here, so if we provide this afterwards, we can bring it. But again, you can find this documentation when you're in the gene curation interface by going to the help drop-down menu and going to help VCI and GCI. That will take you to that document. You want to look on the left-hand side under publishing and approved gene disease record under the GCI specific sections. And you'll find all of those tooltips located to the GCI, as we showed before with those info bubbles. Lastly, for the standard evidence summary text document, we've made some updates along with the SOP. We've added some linked table of contents so that you can quickly get to different areas within the standardized evidence summary text document more quickly. We've also clarified that users may note when a disease mechanism is unknown or unclear. That might be really useful to the end user of that particular record. We have removed language referring to the CNVs not being able to be entered in the GCI because they are now with CA-CNIDs. So you will no longer have to use this language. And obviously, we strongly encourage you to score and use those CA-CNIDs where appropriate. We've rearranged some of the sections to make it hopefully easier for you all as a reader and to be very intuitive approach through the curation workflow. We've also included example text to describe recurations where new information has been evaluated, but the classification has not changed, right? So just being able to give a little bit more clarity for you all. And we've updated and replaced links to examples because, as you know, they start to change their classification with time. And lastly, within the Appendix D is the secondary contributor update. This was a new section we added in the SOP last year, but we also recognized we wanted to give some updates and clarification to the use of secondary contributor because we had a lot of questions. So I want to make clear, like, this attribution should be used to recognize if another expert panel or working group has contributed to your curation. As you all are well aware, there are multiple genes that could have phenotypes arising in different organ systems. And so that means that GSEPs may be interested in similar genes across many of them, not just two. And so we really encourage you all, when this happens, to please use the gene tracker. See what other groups have either pre-curated or have on target to do that particular gene. Reach out to the coordinators of the groups. The Gene Tracker has the email address for all those coordinators. You can click on that coordinator and get that email very quickly. Obviously, you can use the ClinGen website, the group and personnel management, etc. This happens at the stage of the approvals. So again, if you're a biocurator that is in charge of publishing your record rather than a coordinator, make sure you check back in with your GSEP coordinator to make sure whether you need to have an acknowledgement of a secondary contributor. Once you're in that approved classification section, you want to acknowledge the other contributions here. By clicking this, you'll get a drop-down menu in which you can choose whatever group or groups, it could be multiple, that you want to add to that. The other thing I want you to recognize is that if you are doing this and collaborating, is that the GSEP that owns the record in the GCI, there can only be one, right? We can't have duplicate records. So the GSEP owning that record will have to make any updates to the record requested by the other GSEP. This is going to be things like, if you want to update a sentence in the evidence summary, do you actually want case reports added to it? If this is what you're requesting, please be considerate and provide all details for any of the cases to add your evidence summary, sentence, and statements to make it less of a burden and easier for that GSEP to be able to grant your request. We do appreciate if these requests are granted, and if you have any concerns, you want to bring it up with your GSEP chairs and coordinator. They will also in turn bring it up with your clinical domain working group or CDWG chairs and coordinators. Now, I just want to also emphasize with secondary contributor, if you are in there as your affiliation, please do not mark your own affiliation as a secondary contributor. You are the primary contributor and you are already taken care of in the GCI and on the ClinGen website. This is really when you have worked with another expert panel, which could be a GSEP, it could be a working group, or even a variant curation expert panel. You have the ability to acknowledge any of them if they have helped you with the final gene disease validity classification. The secondary contributor is also not needed or required if you're a GSEP that just has plain interest in this gene and you want to publish about it in your manuscript. If you have nothing else you want to add, you have no changes to the classification, you completely accept that gene disease relationship and classification is appropriate, you can remark on any of those genes in your manuscript. We just ask that you give the proper attributions and acknowledgements. List that gene, list the appropriate disease, list the expert panel that did that curation and the link going to that particular curation from the ClinGen website, which will show all of that information. So you don't necessarily need secondary contributor just to say, hey, I'm interested in this gene. It really should be something if you are collaborating with a group to add additional information for that curation. And that is the end of our update. So I'm happy to take any other questions that you may have. Great, that's clear.
I was going to say, just so that we can hopefully get through all of the FAQ questions. I think we had sort of 8 or 9 left from the last meeting. So if there's not burning questions, we will move on. Marina, were you going to share?
Yes, I will
So
share.
just reminding everyone, we started this at the first meeting in September, I think it was. We'd sent out a survey to everyone as part of this group of what were your burning questions of for variant curation, what's confusing in the current protocol, and what do you need clarification on. So we've been working through our answers to those questions, and the slide deck is available, I think, via email. How are we sharing this, Marina? It's not on the website.
Yeah, we can share the slide deck after the fact. I do need host privileges, Kazan, to be able to share my screen.
But I think where we got up to it is we're now moving into thinking about some of the questions that were code specific. So Marina, are you?
Yes, I'm ready.
Okay.
So just 2 quick things before we move on to this. One, thanks to Courtney for presenting all of that. And for the curation reasons, I do want to add that those are also required in the variant curation interface when you publish a curation. They will be the exact same reasons because we wanted to harmonize across the variant curation interface and the gene curation interface. So her presentation is entirely relevant. And it also is critical for our versioning efforts for variant curation. So just wanted to add that you will also see that when you go to publish to the eRepo, that you have to choose that. The other quick thing that I wanted to add as an announcement, I was checking in with ClinGen staff, and actually as of this morning, we have 10 to 15 more spots that opened up in the ClinGen luncheon. So feel free to take this and register if you are coming. We would love for you to join us. So sorry to give you fake news there, but this was added as of this morning. Okay, let's get moving. Okay, so the first code-specific question that we had is around PM2. So the question was, if any subpopulation in NOMAD has a minor allele frequency above the PM2 cutoff, should PM2 not be applied, including the non-continental populations? Because I know that multiple VSEPs will say that only continental populations should count for BA1, BS1. So unlike BA1 or BS1, for most VSEPs, PM2 does usually not exclude any populations, unless the populations have a small number of alleles. So oftentimes they'll say greater than 2,000 alleles. So if a variant's above the PM2 cutoff, PM2 supporting really is what we're talking about here, and any NOMAD subpopulation, PM2 should not be applied. But you should consult your VSEP specifications and your VSEP for clarity there. With the rationale being if it's above a frequency in any population, it's concerning there. Okay, BA1, BS1, and PM2. How do you address poor coverage in NOMAD when applying population codes? And what is the definition of poor coverage? And can data be used for population codes in that case? So NOMAD does have a low coverage flag. I've included a snapshot here. In general, PM2 should not be applied if the coverage is low because you just don't know, is the variant absent or is the variant appearing absent because the coverage is low. My VSEPs personally tend to have coverage concerns if the area is under 30x mean depth of coverage. Usually for the genomes, you're pretty okay that they hover at 30x, whereas the exomes are in blue here are inconsistent and can be very high or very low depending on the area. You'll see this red rectangle here has pretty much absence of coverage except a couple spikes in here. So that would be a region of concern. And you'll see here that this says, warning, this variant is covered in fewer than 50% of individuals in NOMAD. This may indicate a low quality site. Oftentimes we will not apply PM2 supporting in this case. And we'll make a note actually in the evidence summary that coverage is, we're unable to determine absence or presence of the variant because of coverage problems in population level databases. Okay, next question is, what is the intent of the BA1 exception list? Is it that population frequency is not evidence of benignity, or that you can't use BA1 but could use BS1? So per the Ghosh et al. Publication, the BA1 exception list allows a VSEP to titrate their minor allele frequency cutoffs lower without inappropriately classifying known but frequent disease-causing variants as benign. So for example, the hearing loss expert panel has multiple BA1 exclusion variants in GJB2 because we know they're very high frequency in certain subpopulations. Our BA1 cutoff is 0.5%. If we had included those GJB2 variants, we would be closer to like 1 to 2% as our BA1 cutoff. So we are able to titrate it much lower by including those as BA1 exclusion variants. So for BA1 exclusion variants, no benign population codes should be used, as these are often well-known pathogenic variants. For variants that meet BA1 or BS1 because of their frequency, you really should be using case control studies to determine their pathogenicity. So the original use of PS4 that enriched in cases versus controls statistically, instead of using the altered definition of PS4, which is like autosomal dominant case counts, or PM3 case counting, just because of its background frequency in controls. You might just find it by chance. So that's why we need to see statistical enrichment in cases versus controls in many of these exclusion variants. Okay, so Alicia and Jenny.
Yeah. Okay, so our next question wasn't about the population-related codes. We're now moving on to thinking about case observation-related codes. And specifically, the question was about how do we evaluate if PP4 even applies? And are there plans within ClinGen for there to be some HPO-related metrics to be developed, or is it just clinical judgment and diagnostic yield related to the gene? So where we're at the moment is that SVI has recently drafted some guidance around the refined usage of PP4. This unfortunately isn't available as a preprint, but should be published officially shortly. And this guidance is allowing the PP4 strength to be modified relative to the diagnostic yield of a gene. So something that you can ascertain by looking at, say, gene reviews, or as a VSEP, if a gene review for that gene doesn't exist, or if you disagree with it, to come up with that number for yourself. What is the diagnostic yield for your testing methodology for the phenotype observed? And so this box on the bottom right-hand corner is the strength that we've assigned or recommending assigning based on the diagnostic yield for the gene and phenotype. Les Biesecker, who co-chairs SVI, as well as being part of this working group, actually presented on this in quite some detail in our June 22nd call. The recording for that is up online, so you can watch that for more details. But essentially, this box in the bottom right is how PP4 can be applied. The caveat of the current work, though, is that it doesn't address how to determine what constitutes a highly specific phenotype. It is really just focused around diagnostic yield. And so I will hand over to Jenny now, who is going to let us know about some plans that are in the works.
Yeah, thanks, Alicia, and thanks for that update. So just continuing on this question, any plans for some metrics, HPO related or just clinical judgment, and diagnostic yield related to the gene? So Alicia has covered the diagnostic yield related to the gene. So at the current time, I mean, this use of highly specific phenotype really is based on clinical judgment. Although if you look at some VSEPs, if you look at their papers, they have used some statistical methods to look at what is the likelihood that a particular set of clinical symptoms will result in finding a pathogenic variant in that gene. So for example, the platelet disorders group did a really nice statistical study. And then hearing loss have also used this for syndromic hearing loss disorders. So if you want to take a look at those papers, you can see the methods they used. Unfortunately, typically data really isn't available to perform accurate calculations of likelihood that you'll find a pathogenic variant in a gene based on a specific set of clinical symptoms, which is why at the moment we're really just relying on clinical and expert judgment. But this is a question that we've really been thinking about a lot, and it is a really interesting question. So it's something that we're thinking about in terms of maybe using tools like Lyrical and including HBO codes for different clinical symptoms associated with the condition to see if we can help to provide some more guidance on that. This is still in very early stages, so it will likely be a while before we have any answers, but just to let you know that this is something that we are continuing to think about. And yeah, that's all I have for that one, but thanks, that was a great question.
Great, thanks. Okay, so PM1, do you want to take this? I know it's a mix of us, Alicia, but go ahead.
I can start us off and feel free to jump in wherever. So the question here was, can PP3 and PM1 be used together? And if so, is it possible to use PP3 strong and PM1 if at its default strength if Revel is used? So firstly, PM1 is a really complicated code for many reasons. There's not really good guidance for how do we go about defining it. And it's even trickier because it actually represents two lines of evidence concepts in a single code. So it's defined as located in a mutational hotspot and or a critical functional domain without benign variation. And so if we think of those two concepts, the mutational hotspot concept actually overlaps more with the definitions of PM5 and PS1, where we've seen that residue mutated before, but to a different amino acid or a different nucleotide change resulting in the same amino acid change. So in this scenario, if you're wanting to use PM1 in combination with PM5 or PS1, then the combination of those three codes shouldn't exceed strong or 4 points. So you can apply one at strong and not apply the other two. You could do all three of them at supporting. It's really hard to think about in the current framework where we've got these sort of arbitrary strength levels. This will be much easier to combine in the new guidelines when they're eventually released and we're moving to the point system. Or if you're a VSEP that's already using points, you can use these three codes in combination, but they can't exceed 4 points total across them. And then if we think about the other definition of PM1, that it's a constrained, functionally critical region, this concept, of course, overlaps with the evidence that goes into in silico predictors for defining PP3. So in this scenario, if you're wanting to use PM1 on the basis of a constrained, functionally critical region, and you want to use PP3 on top of that, the combination of PM1 and PP3 should not exceed similarly strong or 4 points. And this is also the recommendation that was published in the Pejeva paper, that these two together shouldn't exceed strong or 4 points. But in summary, PM1 is a tricky code because it's combining two concepts, and VSEPs use it. Most of the time, they'll only use it as the mutational hotspot concept or the constrained functionally critical concept, but there are a couple that have both definitions in play at the same time. I think that was it. Verena, did you have any?
Yeah, sure. There's a similar warning of using multiple codes together because it's going through overlapping concepts. So similarly, PVS1 and PP3 should not be used together. So at least within the current guidelines, or things might be upgraded appropriately in the future guidelines. But for example, a Revel prediction for a nonsense variant should not be used in conjunction with the prediction to undergo nonsense-mediated decay for PVS1. A splice AI score for a non-canonical splice variant PP3 should not be used in conjunction with RNA assay data demonstrating an impact on splicing for PVS1. So you wouldn't want to use overlapping codes there. Okay, functional studies versus predictors. We had a question, how can we implement AlphaFold models in the variant curation process? Should they be considered predictions, PP3, BP4, or functional assays, PS3, BS3? It's up to a specific VSEP if they would want to incorporate AlphaFold predictions into their specifications. As their predictions, they should be considered under PP3, BP4, as they do not demonstrate the functional impact of the variant on the protein. But in the new guidelines, they could potentially be scored at a higher level than the current PP3 strong cap. Okay, PS3, BS3.
a question here about functional studies. So can you count multiple assays of the same type? For example, 2 different mouse models. And in general, no, you can't count them additively. Multiple assays of the same type. Animal models are slightly different, and I'll get to that in a moment. But in general, if you just have independent reproductions of what's the same methodology that's examining the same parameters, you can't count this twice. You can't use it additively. Though the variance in those comparable assays can be stacked across the assays to allow the evidence strength to be increased. So for example, rather than just saying it's at PS3 supporting because there's not appropriate controls in that one assay, if you have multiple assays that are the same methodology, examining the same parameters, you can stack controls across the assay in order to increase the strength of that evidence type to say PS3 moderate or even the default strong. If you have evidence by methodologically different assays that are examining different parameters, these can be used additively. So for example, if they both qualify for use at PS3 supporting, you can have 2 lots of PS3 supporting applied, which again is difficult in the current system where we're at strength categories. If you're using a points approach, you would have 2 lots of 1 point, which would add to 2 points equivalent to a PS3 moderate. Animal models here, though, are the exception to that first thing I said, where if it's, well, it's not really exception, but if it's the same methodology, so it's an animal model, but if you have 2 completely independent animal models, so they've been generated through different methods, but they're examining the same variant, and importantly here, we're only thinking about knock-in models. So if you have a nonsense variant in a human, if you just do a general knockout animal model, that's not the variant, so you can't use that as variant level evidence. But if you're doing a knock-in animal model, if you have multiple knock-in animal models generated through different techniques, that data can be used additively. And as a default, it's actually not well covered in the Greenwich paper, but animal models that well replicate the human phenotype can be awarded the default PS3, so at a strong level. And you could combine multiple of them up to an ultimate maximum of PS3 very strong if you have 2 animal models that are perfectly recapitulating the human phenotype.
Okay, PS4 and PM3. We had a question of, would it be worth it to collect clinical databases containing variant and clinical data links in one place, apart from ClinVar, Lovid, or Decipher? So this is for the case level observations. So the VCI has previously surveyed users to ask what resources they would like linked in the curation interface. And based on these surveys, there are currently links to ClinVar and Lovid at the Lovid 3.0 at the variant specific link for each variant, Civic, BRCA Exchange, and LitVar. The Brotman-Beatty Institute database is available with separate documentation for VSEPs only. So please contact your coordinator for that information. As far as development goes, we do need to balance adding resources versus our development tickets. For example, updating the interface for the new guidelines. And we'd be likely to only add those that would be of use to multiple EPs. So if anyone has any suggestions, we'd be happy to take them in the chat, or we have different calls with the coordinators, for example, to receive these suggestions. We also cannot bring in information from access control data, case level databases, because this is pretty much access. As soon you sign up for a variant curation interface login, and everyone has access to the case level information in there, which is why we make you a test to not including PHI in the interface, because everyone can see it. I would say that we also have a DAPC, data access and privacy committee, that works with all of the VSEPs as to how we should collect case level data from labs for internal cases for curation. So that's a little bit separate, as we would not want to include any, we're including minimum necessary information in the variant curation interface because of the PHI reasons I just mentioned. So that is the answer for the case level things. But if anyone can think of something that would be widely useful or applicable to many curators in the interface, we'd love to hear from you there. Okay, so we have 3 minutes. Pam, do you think you would like to chat through, I know we skipped it, your initiation codon variant? Do you think that's enough time?
I mean, I can try, but might be a speedy 2.5 minutes, I think.
We just do the summary slide bit for it, perhaps, or does it need the example to go with it?
It probably doesn't need the example. We can.
Where's the summary slide?
It's slide 30.
Just yes.
Okay.
Okay, so this is basically just in relation to a question of how to deal with start-loss variants, which are notoriously tricky. And there's 2 approaches that one can take, depending on the VSEP's discretion. So the first being, if you have evidence of multiple start-loss variants at that codon, approach 1, you can either use that evidence to upgrade PBS1 moderate, which is the default for start loss, to PBS1, and use it as the default for that gene, with the caveat of there being multiple variants that support the pathogenicity of that start loss variant, and you would explain that rationale in the evidence summary. In the example I used, it was also a functionally critical region, so that was also used to justify bumping it up to PBS1. If you take this approach, you wouldn't want to use PS1 or PM5, as this was technically the type of evidence that was used to bump it up to PBS1. And then one last note on this, as to whether to bump up from moderate to strong or very strong is dependent on the number of variants and the known importance of that region. Approach number 2 is to keep it at a PBS1 moderate and then add an evidence code such as PM5, with the traditional definition of PM5 being listed below. So that was in the past used for missense variants. So we would be using this as the repurposed definition of PM5. So treating it as a variant affecting the same residue as opposed to missense to missense. And so if you were to use this, again, depending on the number of variants, you can treat it in an additive way where it can go from PM5 to PM5 strong. So those are the 2 quick ways of classifying a start loss variant, and it's really dependent on the VSEP, the gene, the region, and all those factors in choosing which one. Okay, 1 p.m. Did it.
Thank you, Pam. Sorry to put you on the spot. I thought it would be useful information to go through. So thank you everyone for attending. I think those were some good pieces of questions and answers of the questions that you gave us.
Yeah, thank you to everyone who submitted a question as well. So this recording will be up online and the slide deck will be made available for people to refer back to. But yeah, that's all we had planned for today. So thanks everyone for coming on, and hope to see many of you next week at ASHG.
Bye.
Bye.